Spring is descending upon New Zealand, and so begins the exam season. Students have a few months until their official external exams, but even as I speak, mock exams are underway. It's a stressful time. There's a lot going on. All of the teenagers of the nation are wondering if they are going to pass the exams and be awarded with entrance to university. I have some experience tutoring for high school exams in New Zealand. I've tutored for English, for chemistry, and for biology. And one thing I always say to students is the best single thing you can do is to read over and try to answer past exams. The worst feeling in the world is thinking in your brain that you're prepared for whatever the test might throw at you, and then being totally blindsided, being asked something that you hadn't even considered, and watching all of your preparation melt away. It feels terrible. But thankfully, past exams are freely available online. And not only are the questions available, but exemplar answers are too. So, the studious teenager can look up the exact standard of the external exam they're going to take, find a past exam from a previous year, and read an exemplar to get an idea of what the markers are looking for, what kind of answer they should try to emulate, what they should try to produce. But then what happens if the exemplar question is just wrong? And not like wrong on a minutiae, like they've missed a point, but like fundamentally wrong. The answer is just factually incorrect. Maybe their reasoning is good, their ideas are good, but the answer is wrong, right? That's okay. I mean, it happens to the best of us in an exam, but it's very dangerous when a wrong answer gets put up as an exemplar for other students to emulate. This is what I think has happened in this exemplar exam. It's for a level 2 biology standard 91157, in which a student is asked to demonstrate understanding of genetic variation and change. A very standard level 2 biological exam topic. The exam is broken down into 3 questions, and this particular answer was given an excellence overall. I presume if you're watching this, you're from New Zealand, but if you're not, in our high schools, we have 4 possible grades. We have not achieved. This is like an American F, I suppose. It's a fail. We have achieved, which is an A, which is the equivalent of like a C, I suppose. You beat the bare minimum. And then we have a merit, which is M. Pretty good. Maybe that's your B. And then we have excellence, an E. And an E is the best mark. So this is an E marked exam. But if we scroll down to the bottom, we can see that the student actually got a merit for the first question, and then 2 excellences for the second and third questions. And to their credit, I've read over the second and third answers briefly, and they seem really good. I have no issue with them. I think an excellence is probably deserved. And even for the first one, I can see why they got a merit. Like, they definitely understand the general concepts. If I was a teacher and I got this exam, I'd be like, wow, my student was paying attention. Amazing. I don't know if this is like a real student or this is just like an example they've made up, but I don't make this video to call out this particular writer. I mean, it's a respectable exam entry. However, in the first question, the one they got a merit for accurately identifying the sex chromosome genotype for a female cat and explaining how the presence of 2 different alleles, F and F, lead to tortoiseshell fur color. And I'm going to claim in this video that in fact they did not do this. They gave a false account of how sex chromosomes lead to tortoiseshell color, even in terms of the exam's framing. An important general piece of advice for an exam is that they are very commonly structured to give you a little story at the beginning. The story will give you some specific information about a particular animal or a particular biological circumstance, and your job will be to use your generalized biological knowledge, your understanding of theoretical concepts and principles, to explain the story that you're told. Now, in this case, and in quite a lot of cases, the story that the exam gives you isn't exactly true. This is not because the examiner is lying to you or trying to trick you. It's just because science is very, very complicated, and we've decided that the way to teach these complex systems is to present a simplified account first, and then layer on the complexity later. Now, I actually hate this idea. I think you should just teach the complexity. And if that means you can impart a narrower but more true picture of the world, then I think that's better. It stops people getting misconceptions. I have a lot of thoughts. I'll save them for another video. For our purposes, we are given the story and we should just take it as true. If we know about the actual reality, great, then that usually will help us understand the question. But if we don't, We just have to parse out what the exam is trying to tell us. The answer that the student has given is wrong in two ways. It's wrong in regards to the reality of how cat gene expression works, and that's okay. This exam is aimed for people in year 12, so that'll be 16 and 17-year-olds. And we don't expect them to have a perfect understanding of cat gene expression. I don't have a perfect understanding of cat gene expression, but they're also wrong in terms of the information the exam has given to us. So what I'm going to do in this video is go through the exam and talk about how I would approach answering it, where they've gone wrong, what the true story is, just to correct the record, because I think it would be tragic to read this exam, try to learn from the answer because it's provided as an exemplar, and then report it, and then be wrong in the exam because you learned from a wrong source. So let's get started, shall we? I have the exam over yonder, and I have a blank PowerPoint slide which I can put stuff on. So we open our exam paper, and here is our story. Some domestic cats exhibit a complete dominance pattern in coat cover. The allele for black fur, uppercase B, is dominant over the allele for brown fur, lowercase b. And so immediately the question is, what is complete dominance? And what is an allele? Now, I know this is about theoretical concepts of genetics, but I think even a basic understanding of the chemical structures, what we're doing when we're talking about genes, makes all of the theory make so much more sense. So let me take you down a little biochemical tangent. Just like any science at a high school level, this is a bit of an oversimplification. DNA, or deoxyribonucleic acid, is an important molecule for life. It acts as the information storage vehicle. Fans of DNA will know it as a double helix-shaped molecule with two parallel strands and then rungs like a ladder going between. Information is actually stored in the rungs of the ladder. So when we are visualizing DNA, it can help to untwist the spiral. Each rung of the ladder is connected by the bond between a pair of nucleotide bases. There are 5 bases: adenine, guanine, thymine, cytosine, and uracil, and they are all pretty similar in chemical construction. We tend to abbreviate them and just call them A, G, T, C, and U. U is a base that replaces thymine in RNA, but for DNA, we don't have to worry about U. We just have these 4. And the rule is pretty simple. G always goes with C, and T always goes with A. Now, a very good question that you might ask is, how the heck does this, the sequence of 4 bases, create a human, right? How does that create an animal? How does that make any kind of life? And the answer to that is very, very complicated, but it starts by converting the information stored in the DNA into a physical structure. What we do is we unwind the DNA, we use a strand of RNA, which is similar to DNA in structure, but only has one strand. We peel one of the 2 strands away, we put a piece of RNA on with the complementary pairs. So if we peeled away the top layer of this, we would then put a piece of RNA on with a G and an A for that T and an A for that T and so on. And then we would send the RNA off to be converted into a protein. Thankfully, there's a handy translator. The way the protein construction works is that the DNA is read in sets of 3 letters called a codon. So here we have 2 codons worth of information. Codon 1 and codon 2. Each of these codons tells the ribosome, which is a little biological machine thing, which amino acid to stick onto the end of the protein. So an amino acid is just a little molecule, and it has an arm on either side that can bond to a previous amino acid and a future amino acid. And proteins are just lots of amino acids chained together. The codon tells the ribosome which amino acid next to add to the chain. So the top sequence of this first codon reads GAA. If we come down to this codon table, we can find GAA here, and that is associated with the amino acid GLU. The 3-letter names are all abbreviations. So when you put the sequence through the ribosome, it'll staple on first a glutamic acid, and then this next one, TCA. Now we're feeding RNA into the machine, not DNA, so the T gets converted to a U, and we end up with UCA, which codes for serine. So we go glutamic acid, serine, and then we'd keep reading off until we had a long chain of amino acids. Now there are a few important features of the chain. The first is that AUG, this one that's highlighted in red, MET, is the start codon. So the first amino acid in a protein is always MET, which is short for methionine, and that tells the little biological structure that's reading down the DNA sequence where to start making the protein. Once the protein has started, it'll keep going until it hits either UAA, UAG, or UGA, in which case it will stop, and the finished protein will pop out and be ready. And now that we understand how a protein is constructed, we can understand what a gene is. A gene is a section of DNA that codes for a single protein structure. It creates one of these long-chain macromolecules that do most of the important stuff in our bodies. Usually a gene will have a start codon and an end codon, and a long sequence in between. There's also other stuff. There'll be a regulatory structure up here that controls how the gene is expressed, but we can think of the gene for the purposes of this exam as just the area between the start codon and the end codon. And what it's going to do is make a protein. So what does it mean then to say that you have the same gene as someone else? That means that you and that other person both have a sequence in your DNA that codes for a protein that does the same thing. The gene might make a pigment that gives your hair or your eyes or your skin a color. It might make an enzyme that helps you break down your food. All humans more or less have the same genes. So what then creates the great diversity that we have? Well, the diversity that we see in humans is because of alleles. An allele, in simple terms, is a version of a gene. So we might have two alleles, allele A and allele B, and they're in the same place in the chromosome, and they do the same thing. If the body needs to make an enzyme to break down a particular sugar, it'll go to that place in the chromosome and read off that gene. Alleles are different in that some number of their bases, their letters, are different from each other. So allele A might have a TA in this position, allele B might be the same over here, but then this TA is replaced with a GC. If you change out a few amino acids in a protein that's thousands of amino acids long, it's not going to radically change the function of the protein. The function of a protein is just controlled by its shape. They're kind of like little machines. So if you change a couple of amino acids, what you're going to do is give the protein a slightly different shape or the ability to form slightly different bonds with certain other chemicals. And now we can loop back to the exam question. The allele for black fur, which we're going to call B, is dominant over the allele for brown fur. Now, there's a further catch. We have 23 pairs of chromosomes. We get 23 from our mother and 23 from our father. They're called pairs because the chromosome one that you get from your mother has the same genes on it as the chromosome you get from your father, but it could have different alleles. So they are more or less doing the same thing. If you go to a particular place on them, you'll find a region that codes for a particular protein, but those proteins could be slightly different from each other. Now this is the case for all of our genes. We have a pair, and so we have two alleles in our body for every single gene, at every single position on the chromosome. Okay, so what does it mean to say that one gene, in this case black fur, is dominant over the other gene? Well, if both genes are read off, the creature, the organism, is going to present as only having the dominant gene. One thing I remember being confused about when I was first learning this is what makes a gene dominant or recessive? Is there some letter you can look at that's tells the body this gene is going to be dominant or this gene's going to be recessive, and then the body kind of knows to make it recessive? And the answer is that's not really how it works. In fact, true complete dominance and recessive relationships are very rare and usually happen because one gene is just broken. The body is not going to discriminate. It's just going to read off both genes. But imagine that this one here has a mutation in it that makes this a CG, and that causes the whole thing to break. It just doesn't read a protein anymore. This can happen if the start codon mutates, and then the body will just be unable to start making the protein because the start codon is broken. Or it can happen if you added a letter in here, and then the entire protein got shifted down, and then all of the letters just became nonsense. You just made a random protein squiggle that didn't have any effect. In that case, this would be an allele, but the allele would just do nothing. And the fact that it does nothing would make it a recessive allele if the body could just use the other gene whenever it needed to do the thing. So say you were looking for some pigment to make your hair black. A cell that needed the pigment would go to the gene and start reading it off. And if it read off this top gene, well, it wouldn't do anything. But also it wouldn't make the pigment, so the cell would keep needing the pigment, and then eventually it would start reading off the other gene, and then the pigment would come in the cell, and the cell would be like, I don't need a pigment anymore, and it would stop. And so if you had two working genes that were the same, you would end up with a bunch of pigment, but if you had one broken one and one working one, you'd still end up with a bunch of pigment at the end, because the cell would just stop when it got what it needed. However, if both of your genes were broken, then you could read off till the end of time and you'd never get the pigment, because you just didn't have the ability to create it. And so you would end up with hair that had no pigment. I guess you'd end up with white hair. And we'd call that a recessive trait. But it's not because, like, this gene, like, bows before the other gene. It's just because, the gene's broken. And so in order to really tell the story of dominance and recessive, or codominance, as we'll get to, you really need to know the particular story of what's happening in the body. Usually the explanation for why something is recessive is just like a pretty simple, like, mechanical story of something not working right. So, to apply that to the exam, the allele for black fur, uppercase B, is dominant over the allele for brown fur, lowercase b. What that means is that if you have one gene that codes for brown fur and one gene that codes for black fur, you have one gene that's going to make a brown pigment and one that's going to make a black pigment. The black pigment is just going to overrule the brown pigment. You put both in the hair, but the hair's just going to turn black because the black pigment takes over. So the black fur gene is dominant, and the only way to get brown fur is to have two brown fur genes, so that no matter which gene the body goes to get pigments from, it's going to get brown. Let's move on with the question. The gene for tail length is not linked and is located on a different chromosome from the coat color gene. So what's linkage? Linkage is where two genes are on the same chromosome. So as I said, we have 23 pairs of chromosomes. A chromosome is a long, long strand of DNA that contains lots of genes, so lots of start and stop codons with spaces in between, and can make lots of different proteins depending on where you start. When you inherit a chromosome, you inherit the entire chromosome, with some exceptions that aren't too relevant for this question. What that means is that if two genes are on the same chromosome, if you inherit one, you also inherit the other one. This is a very interesting novelty of evolutionary biology. That means that if you evolve some gene that's really great, it's not just the gene itself that gets a selective advantage, it's all the genes around the advantaged gene also get boosted in their fitness just by virtue of being right next to a really good, powerful gene. Hi, Abel. This is Abel. He's a cat. I don't know if you can see him very well on the camera, but he's here to demonstrate what a tabby cat looks like. You have both brown and black fur. Isn't that crazy? Because you're a tabby. The story doesn't account for that. Yeah, you got brown and black fur. It's crazy. Who would have expected it? So we're told that tail length and fur color are not linked on the chromosomes, which means that when you are inheriting those two traits from your parents, whether you get black fur or brown fur has no impact on whether you have a short or long tail, because each of the chromosomes is divided up in a separate action. We're told that the allele for long tails, capital L, is dominant to the allele for short tails, lowercase l. A cat that is homozygous for black fur and long tail is crossed with a cat that is homozygous for brown fur and short tail. And the first question that we're asked is, specify the genotype of individuals in the F1 generation produced by this cross. And the student has given the perfect answer. It is BBLL. We know the answer to this because we are told that both cats are homozygous. That means that at the same gene on each of their paired chromosomes, they have the same allele. Now this is quite convenient for us because it means that it doesn't matter which allele they pass down. There's a 50% chance that parent 1 passes down the first B and a 50% chance they pass down the second B, but the kid's always getting a capital B. And same for parent 2, but the kid's always getting a lowercase b. What that means is that in the first generation, the child always gets a uppercase B from one parent and a lowercase b from the other, and they always get a uppercase L from one parent and a lowercase l from the other. It gets more complicated in the next generation, and that's where the punnet square comes in. Now this is slightly more complicated than a typical punnet square because we are doing a dihybrid cross. We are told, use the punnet square below to show the F1 gametes resulting from the cross and the possible genotypes within the F2 generation of cats. So the question wants us to recall, how does a gamete work? What happens when you produce a gamete? And the answer is that you end up with a cell that has half the usual number of chromosomes. We don't spend very long in our gamete phase. As mammals, we create sperm and egg cells, and then we very quickly have a fertilization event and go back to having the normal number of chromosomes by combining. But the first question we have to ask is, what kind of gametes would one of these first-generation offspring produce? So we have the genotype, and we're going to independently and randomly pick one of the first two alleles to give to the offspring, and one of the second two alleles. So we could pick a capital and a capital, or we could pick a lowercase and a capital, or we could pick both lowercase. Now remember, we're only picking one, so we're not going to pick like two uppercase B's, or an uppercase L and a lowercase l, because it's a gamete. So it only has one of each allele instead of two. So we might have B from here and L from here, or B, L, and so on. If we look at the answer, they've done it perfectly. We have 4 possibilities in the F1 gametes on the horizontal row and on the vertical column. And all we have to do to construct the Punnett square is to combine the gametes. So combining this gamete with this gamete, we just get all capitals. We get 4 capitals. And combining the lowercase gamete with the other lowercase gamete, we get the lowercase offspring. Cool. So now we have 16 different genotype possibilities. Some of them are the same but rearranged. So, for example, this one here, BBLL, is identical to this one here. It's just that the first individual is getting its capital L from this gamete, and the second is getting its capital L from this one. But they will look like the same kind of cat, because they have the same overall structure. So far, students doing amazingly. Exemplary exam. Very good. Question C. Give me expected phenotype ratio resulting from the cross, indicating the phenotype each value represents. Again, the students done great. They've answered perfectly. The ratio is going to be 9 to 3 to 3 to 1. This is a typical dihybrid cross ratio, but we can go up and have a look at the Punnett square to see exactly what's going on. So remember, the rule is capital letters are dominant in this case. So if you have at least one capital letter, you are expressing the dominant phenotype. Every cat with at least one capital B and at least one capital L is going to be a black, long-tailed cat. So let's just see how many we have. All of the cats that come from this top row gamete are going to be black, long-tailed cats, because the gamete has both dominant alleles, so it doesn't even matter what the other gamete brings to the table. This gamete alone is going to control the phenotype. Same for this one in the column. In addition to those 7, we have 2 more. We have this guy and this guy, giving us a total of 9. Now we can start looking at the recessive possibilities. The first is that you have a capital B, but not a capital L. This will make you a black cat, but with a short tail. There are 3 cats that fit this mold, and they are these 3. We can also do the opposite. You have a capital L, but not a capital B. You are a brown, long-tailed cat. That accounts for these 3. And then all that remains is the double recessive cat that was lucky enough to have 2 gametes that had double recessive alleles. That single individual is going to be the double recessive brown, short-tailed cat. And that's our Punnett square. It looks kind of arcane when you look at it, but just thinking it through and running it through in your head, it's not too hard to come to the correct conclusion. Here's where it all goes wrong. Again, I'll read through the question and give some context, and then we'll have a look at the student's answer and see why it's so wrong and why it annoyed me enough to make me want to make this video. So, here's the question. Some cats have fur color that is an example of codominance and sex linkage. So let's talk about what those two things are. In a classical dominant-recessive relationship, if you have the dominant is fully expressed. This is the case we talked about earlier, where maybe the recessive is just a broken gene, and so the recessive trait is not being able to do the thing that the gene would normally allow you to do. But what happens if both genes make a protein successfully, but the proteins have different features? They are different colors, they break things down in slightly different ways, or they bind with more or less efficiency to different substrates. In that case, you might get codominance. In this case, when the body needs a particular protein and it starts reading through the genome, it'll create some of each of the proteins. And so in a codominant system, you can have this one single gene being expressed at the same time, but allowing you to do two different things because you have some of each of the protein in your body. Or in the case of something like a pigment, you could have half and half of each pigment, which would produce a phenotype, a physical appearance of having a kind of blend of the two colors, of having an intermediary feature. Codominance is actually very common in genetics. It's more common to have this kind of system where you have two slightly different proteins being produced than the true recessive situation where the recessive phenotype does nothing. In the dominant recessive situation, you only have two phenotypes. You have the dominant phenotype and the recessive phenotype. But in the codominant situation, you have three phenotypes. If the two codominant genes were capital A and lowercase a, you could have the double capital A phenotype and the double lowercase a phenotype, and those would be different. But having one of each would be a third different phenotype. Maybe it would be in the middle between the other two. And in fact, in nature, oftentimes you have multiple genes that are spaced out across multiple chromosomes, all contributing to a trait. So there are lots of genes that control how tall a human is going to grow, and the phenotypic height that you have is a kind of codominant average expression of all of the alleles across all of these different genes. But we don't have to worry about that because we're just talking about single gene alleles. The other thing that was brought up was sex linkage. Now this is kind of a skill check, this is a stat check. Have you remembered how sex chromosomes work in vertebrates? There are two different types and they have completely different genes on them. So the X chromosome is pretty long, it's got quite a lot of stuff on it, and the Y chromosome is tiny. The main thing that it does is create proteins that are regulators that will change the way that genes on the X chromosome are expressed. So if you make a bunch of these regulatory compounds from the Y chromosome, they will go over to the X chromosome and start changing the way and the times and the places that the X chromosome is expressing itself. Now, because the Y chromosome is so small, the X chromosome is actually kind of self-sufficient. You don't need to have a pair of them in your body to get the function of it. It's totally possible as human to be X0, just have the one X chromosome and nothing. It will have some effect on you. You'll probably tend towards some differences in sex characteristics and this kind of thing, but you'll be a perfectly normal human, which is not the case if, say, one of your two chromosome 1s were missing. You were like chromosome 1 and then nothing. That is not a viable human. That human doesn't make it to birth. That human fails as a very young ball of cells. But with sex chromosomes, it's kind of okay. You can even have duplications. You can be XY. That's stable. You'll kind of end up intersex. You'll have some female-associated traits and some male-associated traits, but again, it's a perfectly normal state of being for a human. So the sex chromosomes are pretty fluid in comparison to all the other chromosomes. They're inherited in the same way as the other chromosomes, though. And in the sort of archetypical biological cases, although of course there are many exceptions, if you have two X chromosomes, you are a female, and if you have an X and a Y, you are a male. You can imagine how the inheritance works. The mother, who has two X chromosomes, is always passing down an X chromosome. So no matter what, you get an X chromosome from your mother, but your father has a 50% chance of giving you a X chromosome and a 50% chance of giving you a Y chromosome. If you get the X, then the offspring will have two X chromosomes and be a female. If you get the Y, the offspring will have XY and will be a male. So the father's gamete, the paternal gamete, determines the sex of the offspring. This is the archetypical case of why Henry VIII was wrong to execute his wives for not giving him sons. It was actually his sperm that was giving him sons. He was being portrayed by his own gonads, and the woman had nothing to do with it. They were always giving an X chromosome. It wasn't their fault. It was Henry VIII's fault, but he didn't know that, and so he instead murdered his wives. Okay, so that's the story with sex chromosomes, and a sex-linked trait is a trait that's on the X chromosome. It's going to be expressed differently based on whether you are a male or a female. So, in cats with orange fur, pheomelanin, orange pigment, completely replaces eumelanin, black or brown pigment. So we have the secondary pigment, and if you have the ability to produce it, it will override the black pigment. The gene to produce pheomelanin is on the X chromosome, so it's sex-linked. The orange fur allele is capital F and is codominant with non-orange. Okay, so this is like a bit confusing, right? Because if you have the F, then you override the black or brown, you override the B's from earlier. But if you have the lowercase f, which means that you're either an intermediary form or both black and brown are being expressed, males can typically only be orange or non-orange. The allele has a codominance pattern if you have both F and lowercase f, but males don't display that codominant phenotype. They only display the full orange phenotype, or the not orange. We're told that sex determination in cats is the same as it is in humans. And this is actually important, although beyond the scope of a typical high school science exam. And it's important because not all animals determine their sex in this way. And even animals that are like, big animals, big vertebrates, warm-blooded, can do it quite differently. For example, birds. Birds have a similar system where they have one sex that has two of the same chromosome and one that has a sexual difference chromosome. But they use Z and W, which are named differently to reflect their slightly different function. If you have two Zs, you are a male bird. And if you have a Z and a W, you're a female bird. This is the opposite to the human case, where if you have two X's, you're a female, and a bird, if you have two Zs, you're a male. But we don't have to worry about this because cats are mammals and they do it the same way as people. They have X and Y sex chromosomes. Female cats can have orange fur, fur without any orange, or tortoiseshell fur. See image on the right, which is not there, in which some parts of the fur are orange and others are non-orange. And so this is the codominant phenotype, which is good because we've already been told that it's codominant. Now we know how the codominant phenotype looks. It's not a blending. It's not like you're kind of like orangey brown, except you have patches of orange and patches of black or brown spread out around you. So in some places of your body, some parts of your skin, the hair that grows will be orange using the orange phenotype. In other parts, it will be using the black or brown phenotype. And we're told that only occurs in female cats. Some cat diseases are known to be sex-linked as well. Male cats have been found more susceptible to recessive sex-linked diseases than female cats. And finally we get to the question, I know it's been a while, finally we get to the question that drove me to make this video. Question D. Evaluate the inheritance patterns of cats to include complete dominance, codominance, and sex linkage. In your answer, refer to the examples above and include a discussion of the two patterns of dominance, why only female cats can have tortoiseshell fur colour, and the similarities and differences of recessive and dominant sex-linked genes. I'm going to go through the student's answer, give my thoughts on it, give how I would correct it. If there's anything missing, if we need to go back and answer some extra bits, we can do that. We can do that at the end. So, the student begins by writing, the first pattern of dominance within cats is codominance. Codominance is where there are two dominant alleles, and when there is a heterozygous individual, both phenotypes will be displayed, brackets, each cell can display a different dominant gene. And that's perfect. So this is an amazing answer. We're off to a great start. The second pattern is complete dominance, where the pheomelanin orange pigment completely masks the eumelanin, meaning any cat with the orange pigment allele will display the orange phenotype. And this is wrong, but not that badly wrong. The reason it's wrong is because the gene that codes for brown pigment and the gene that codes for orange pigment are not different alleles of the same gene. They're different genes. And so, as we can see with the tortoiseshell cats, it's not the case that if you have the orange pigment gene, you are never going to have any brown phenotype showing, right? In the case of the F heterozygote, you have the orange gene being expressed, but not overwhelming the brown gene. The reason this is confusing, though, and it's not necessarily the student's fault, is because there's another process going on with the tortoiseshell pattern, which we'll get to. If you want an example of complete dominance in a cat, all you have to do is go back to the prior questions, right? We know that black is dominant over brown. We know that long tail is dominant over short tail. If you have those alleles in any such way, you're never going to have the phenotype of the recessive trait. Whereas merely having a dominant F allele is not enough to suppress completely the brown phenotype. But you can see where the student is coming from, right? Because it is true that the F allele does suppress the phenotypic expression of the black and brown alleles on the other gene, but this isn't a simple dominance relationship because they are not alleles of the same gene. It's a more complicated setting, but I can see why you'd want to call it dominance. Any cat with the orange pigment allele will display the orange phenotype. This is more common in males. No, it's not necessarily true that it's more common in males. It's just true that the males can't display the heterozygous phenotype, but that's okay. It's a minor thing. Next, sex linkage is where there are alleles coding for a particular trait carried on the sex chromosome. However, this doesn't change the 50-50 possibilities for each sex. Okay, so that's an astute statement. Remember, you always inherit an X chromosome from your mother. So whether you're a male or a female, you have a 50% chance of getting one of your mother's sex chromosomes and a 50% chance of getting the other. So in that sense, it doesn't change the probability of which one you're going to get. For the other X chromosome, the one that you get from your father, you only ever had one option of X chromosome to get, but you had a 50% chance of being a male, and that would have just been your phenotype instead. It just means that only females, 2 X chromosomes, can have tortoiseshell fur because the gene for black or brown pigment is only found on the X chromosome, which males only have one of. This is not true. We know it's not true because we actually are working with two different genes. This gene, the BB gene, is not sex-linked. It's not on a sex chromosome. It is on a normal chromosome and inherited and passed down independently of sex. So males can actually totally have two black fur genes, 2 brown fur genes. That's fine. It's the F gene, the orange or not orange gene, is a second gene which is sex-linked, which if you have the capital F version, overrides the black or brown gene on the other chromosome. This black or brown pigment, continues the student, is a recessive allele, meaning it is only produced in the phenotype when it is present. Now you can see the mistake they've made, and it's a very innocuous mistake, easy to make under exam conditions, right? They have understood this F and F relationship as separate from the previous information that we were given. You can imagine like maybe there's like 2 different species of cat, and one of them has the BB version, but that's not the case. We're talking about domestic cats. They all have the same genome. Okay, so the black or brown pigment is not a recessive allele. Brown is a recessive allele, but black is dominant. And the second part, that it is only produced in the phenotype when it is present twice, is definitely not the case. So it's not the case at all that something about having 2 copies of the recessive allele is what makes the recessive allele activate. It's the case that having any copies of the dominant one negates the recessive. Having just the recessive and then nothing else is going to give you the recessive trait because you don't have the dominant, so you've only got the recessive. You have the recessive trait. This is not much of a problem for like a human, where in our body we have linked chromosomes for everything except the sex-linked genes. But if you're, for example, a parasitoid wasp, then you have a different system. Parasitoid wasps have a feature called haplodiploidy. So diploidy is the property of having pairs of chromosomes, and haploidy is the property of having single chromosomes. So we are diploid, and our gametes, our sperm and eggs, are haploid until they recombine into a diploid zygote. Now, parasitoids do it a bit differently. Female parasitoids are diploid. They have pairs of chromosomes just like we do. However, wasps have the ability to control whether they make males or females. So if you think of like a bee hive, you have the queen bee, and she's always a female, and then you have the worker bees, and they're all females as well. And then you have the drones, the male bees, and their only real job is to mate and then die. They don't live very long, they don't help at all, they're kind of useless. So the queen bee, who wants workers for the hive most of the year until it comes to reproductive season, wants to make females. So, okay, the queen is going to decide whether to make a male offspring or a female offspring. How does she do it? Well, to make a male bee, the female doesn't have to mate with anybody. All she does is she lays an egg just by herself, and she gives the egg either her first chromosome or her second chromosome. And that means that the male wasp, the male bee, bees are a kind of wasp, in fact, is haploid. It's only going to have either A or lowercase a, but it doesn't have a father at all. Male wasps do not have fathers, they only have mothers. You can think of them as like half clones of the mother. When a queen wants to make a female bee, a female wasp, she'll mate with a male. The male will just pass down all of his chromosomes, because he's only got one set, so he just always passes down the same chromosomes. The female will pass down one of hers, and you'll get a baby female bee, who will have one set of chromosomes from her father and one set from her mother, but every time this male bee mates, it'll always pass down the exact same set of chromosomes. There's no, like, random assortment. It's just always the same set, because he only has one set, because he doesn't have a father. Now, the way bees work is that they will usually mate only a few times, but they have internal organs for storing sperm, and when they mate with a male, they will gather so much sperm that they'll be able to fertilize thousands and thousands of females. So they just have this pool sitting inside them that they can activate to add a second chromosome to the offspring and change it from a male to a female. It's very, very fascinating. Very different to a human system. The reason I bring all this up is because if you're a male wasp and you have a recessive trait, you have the recessive trait. You only have one chromosome set. There's no room for a dominant structure. In the female wasp, they could have that recessive trait and then have a dominant one to mask it. So they could be dominant A, lowercase a, have both, and then express the A. But if the male wasp has the lowercase recessive trait, it's getting hit by the recessiveness. Like there's no masking it. It's not that it needs two of those recessive alleles to be recessive. No, he's getting all the recessiveness. Maybe that's why they die so quickly. The same is true for us on our sex chromosomes. If you're a male with a recessive trait, and it's on the sex chromosome, it's on the X chromosome, and you've only got one, and the Y chromosome doesn't have any genes on it, and you've got that one recessive trait, you've got nothing to mask it, you're kind of screwed, right? You're getting hit by the full brunt of the recessiveness. So this statement, it is only produced in the phenotype when it's present twice, is completely wrong. I think it's relevant that it's completely wrong because the rest of the essay is going to be built on this misconception. Okay, next sentence. As it's only found on the X chromosome, only female cats can display it, as they must have two recessive genotype, black or brown, on two X chromosomes, e.g., XX both have the recessive trait. This is not the case, right? The female that has the lowercase f twice and the male that has a single copy of the lowercase f will have the same phenotype from that gene. In both cases, those cats don't have a ginger allele, and so the other gene on the non-sex-linked chromosome is going to either give them black if they've got the dominant black gene, or brown if they've got recessive brown. But that's a very different story to what the essay is presenting. Males cannot, however, because even if they have one recessive allele, X, the other is a Y chromosome, meaning it will mask the recessive trait and will not be displayed in the phenotype. Now, this is not the case. The Y chromosome, the main thing it can't do is mask X chromosome genes because it has different genes on it to the X chromosome. Different genes, they do different things. We can't group them up. We can't do the pairs of letters like we can with the other chromosomes because they don't have the same genes on them. So the Y chromosome provides no masking. It does exactly the opposite of what's being claimed here. Only females that have the genotype capital F lowercase f will have tortoiseshell colours, where F is recessive and F is codominant. Recessive won't be masked, but it will still have the orange pigment as well as the black-brown tortoiseshell. This is also very wrong. We are told explicitly up here that the orange fur allele F is codominant with non-orange F. Dominance is not a trait of an allele. So it's not that capital F is a dominant allele and it's that's its trait. Dominance is a relationship. So capital F could be dominant or recessive in regards to lowercase f. It's not a trait of an allele that can come out without a second thing to have the relationship with. So Codominance is the relationship that we are told we have between the orange and non-orange alleles. So lowercase f can't be recessive. Now, there is a recessive component to the system, but that's happening in the B alleles, on the other gene, on the other chromosome. It's also a total contradiction to say that the recessive won't be masked. The definition of a recessive gene is a gene that's masked by a dominant gene. So what actually is happening? What's the codominance relationship? Well, in some places on the cat, capital F ginger gene is being expressed, and in other cases, the lowercase gene, F, is being expressed. And in fact, it's approximately 50% of the cat that's getting each gene. Why the heck is that happening? What's the underlying system? Well, interesting question, hypothetical viewer. The answer is a really weird thing that happens in mammals called X inactivation. We've been kind of going along with the story that females have two X chromosomes and they have both and they're working like a normal gene, a non-sex gene. But in fact, what happens is that somewhere in development, pretty early on, when you're just a ball called a blastocyst, when you're just a guy that looks like this, you're kind of a clump of cells. You have maybe 100 to 200 cells, or just a little kind of clump of cells very early on in development. When you're in the blastocyst stage, each of your cells will randomly choose to deactivate one of its two X chromosomes. It'll turn it off, it'll neutralize it, and turn it into something called a bar body, which is like a dead chromosome. It's a chromosome that doesn't do anything. And so what happens, in fact, is that from that point on, that cell and all of the descendants of that cells have one working X chromosome, and the other is doing nothing. If you take an adult mammal, an adult human who's female, and look inside their cells, they will still have this bar body, this deactivated X chromosome, and it won't be doing anything. This is why you can get away with having only one X chromosome in you and having like X0. It's okay, because in fact, in adult female mammals, you only have one X chromosome that's actually doing anything anyway. The trick is, though, each of these cells in the blastocyst independently and randomly selects one of the two chromosomes, one of the two X chromosomes to deactivate, and they don't all have to choose the same one. So, the adult mammal is a mosaic of the daughters of the blastocyst cells that chose the first chromosome and the daughters that chose the second. And now you might be starting to see how you get a tortoiseshell cat. Remember, the tortoiseshell cat is heterozygous in the sex chromosome for either having a ginger color that takes over from the black or brown color, or having the black or brown color. So, for example, this X chromosome here might be uppercase F, and this one here might be lowercase f. Okay, but we're randomly turning one or the other one off. So 50% of the cat has this chromosome here turned into a bar body, it does nothing, and is only expressing this chromosome, which has the recessive allele. So from all of those cells, it's just producing the black or brown cat. However, the other half of the cat is only producing the orange allele. In each of those individual cells, the case for the male and the female is actually identical. They both have one working X chromosome. It either has the capital F or the lowercase f, and that is going to determine entirely which color of hair that cell produces. It's just that the heterozygous female is made up of two halves of a blastocyst that went different directions way back in the day. This is very complicated and beyond the pay grade of the average level 2 NCEA student. So we cannot fault the student for not knowing about this strange bar body heterozygous relationship. However, I think we can fault the exam question writers a little bit for writing a question about this situation without explaining that it's possible for this weird codominance trait to exist. But even if we didn't know this, even if all we had was the case we're given in the exam, it's still the case that the student's essay is wrong. All of this stuff about the Y chromosome masking the X chromosome is not true. The stuff about the the lowercase f being recessive when it's a codominant relationship is not true. Let's read on, shall we? However, a male doesn't have two X chromosomes, so recessive eumelanin will always be masked. Now we know that this is not true. The student knows this is not true because they know that there are black and brown cats. A recessive sex-linked gene likely decreases genetic diversity because it is not often expressed. This is also false. That's because genetic diversity doesn't rely on phenotype. Genetic diversity is just the diversity of alleles that an animal has within the population. Species might have high genetic diversity if for any given gene there are lots of different alleles, or if there's only one allele for all the genes because they got really bottlenecked, they'd have low genetic diversity. If the genes aren't expressed, right, if you have a bunch of these genes that are there in the population but not expressed, it still counts as genetic diversity, it's just not phenotypic diversity. Moving on, must have two copies, double X, chromosomes to be visible in phenotype. Again, not true. In fact, all of the X chromosomes that we're working with because of the bar body thing are sole chromosomes. You only ever have one activated in any given cell, and the males only have one in their whole body, and it still has a major role in phenotype. Sex-linked diseases can be very harmful to a population. Male cats have been found to be more susceptible to sex-linked diseases than females, meaning this disease is likely found on the Y chromosome and only inherited by males. This is an interesting idea, and it makes intuitive sense, because only the males have the Y chromosome, so surely that's where the sex-linked diseases would be. I imagine that it's at least possible that there are sex-linked diseases on the Y chromosome, but it's not very common. And the reason it's not very common is because there's almost nothing on the Y chromosome. It has hardly any genes at all. A lot of the sex-linked diseases are recessive diseases carried on the X chromosome. If you have a recessive trait on the X chromosome, a male, if it gets a copy of it at all, is screwed. But a female, if it gets a copy of it and the dominant allele, well, as long as the cells that haven't turned their dominant allele into a bar body can export the dominant thing to the rest of the body, they can make up for having the recessive allele. And so the male is much more likely to be affected by the recessive trait. An excellent case of this is male colorblindness. So colorblindness is a recessive sex-linked trait. It's on the X chromosome, and you won't be colorblind if you have at least one X chromosome with the gene that makes you not colorblind. So if you're a woman and you have a colorblindness allele and a not colorblindness allele, you're fine. But if you're a man and you just have the one colorblindness allele, then you're out of luck. Women can still be colorblind, but they need to have both. They need to have a colorblindness gene from their mother and their father. And because the father only has one X chromosome, like they're either given that X chromosome or a Y, the only time a female mammal can be colorblind is if their father was colorblind himself. But it's very easy for a female to be a carrier of colorblindness and have male children who are colorblind without being colorblind themselves. Anyway, I digress. Let's move on with the show. Or, more likely, the sex-linked disease is a codominant allele which is only sometimes expressed in males and rarely in females. So this doesn't really make sense. We know that if it's a codominant allele, males can't display the intermediate phenotype, and if a disease is a codominant allele, then we'd expect having the disease would at least give you some part of the disease. So this statement is at least, I think, needing further clarification. Next, dominant sex-linked genes are likely to always be inherited, unless on the Y chromosomes. Then, only males would always get that allele. So this is, again, a bit confused because there's almost no genes on the Y chromosome. So a male is still getting most of their sex-linked traits from their X chromosome. I also don't know what they mean when they say dominant sex-linked genes are likely to always be inherited. Remember, a gene is not dominant. An allele is dominant within a gene. A gene itself isn't dominant. It's not a property of a gene. But even if this person had said dominant sex-linked alleles are likely to always be inherited, that still doesn't follow, right? We dominance and recessiveness have no impact on the likelihood of inheritance, they only have an impact on the phenotype of the offspring. However, continues the writer, a recessive sex-linked gene only gets expressed in the phenotype if it is displayed twice. Again, not true. Recessive traits are happily displayed with only one recessive allele, so recessive genes are more likely to be on the X chromosomes. Brackets would never be expressed on the Y chromosome, as X would always mask it. I'm trying to pass this out. A recessive gene only gets expressed in the phenotype if it is displayed twice. So, recessive genes are more likely to be on the X chromosomes. Right. Okay, so the student is under the false assumption that if you have a recessive allele, it's only expressed if you have two of them. And I can see why they are under that assumption, because when you're taught about recessive alleles, you are taught that you have to have two copies to have the recessive trait expressed. However, again, it's not the having of the two that make the recessive trait expressed, it's the lack of the dominant one. When the student says that a Y trait could never be recessive, they say that because you could never have like two recessive genes on Y chromosomes because you can't have two Y chromosomes. And if you're under the assumption that you need two recessive traits in order to have a recessive phenotype, then that's actually a logical conclusion. So I kind of respect this statement because although the student has made a past misconception, you can see that they're turning the logic around in their brain and building out the conclusion. It's just that the conclusion is very confused, which is very unfortunate. And I can see why you get a merit, because the student is displaying like good critical thinking processes. It's just that it's a terrible exemplar, because who knows, it's all wrong, unfortunately. Final sentence. The similarities is that they both don't affect the gender ratio of the offspring and could both have positive and negative consequences to the offspring. Okay, sure, that's like a concluding statement. It's very vague. I'm not sure if I have much to say about it. But that is the merit level answer in this excellence exemplar. If we come back up to the bullet points, that last sentence was, I think, a quick way to try to get a mark for the third bullet point, but it's a bit too vague to get a tick. And yeah, so that is the entire question. I hope this doesn't seem like a mean-spirited attack on this essay, because it really isn't meant to be. I'm only annoyed at like the marker, I suppose, for not catching any of this, and the fact that this is put up as the first exemplar for this standard. I know that like it's not a big deal, and if you're a student, but like if you're a student and you're looking for an excellence exemplar, this is kind of like all you get. It can be really hard as a student to figure out what examiners want from you. You get a knack for it over time, and especially if you have been in academia for as long as I have, like you start to be able to read the intentions of the examiner. But when you're young and you've never done an exam before, sometimes you just don't know what they want, and you just write something and it's not what the examiner wants. So, like as a tutor, having passed exams with exemplars is critical for teaching people about this new experience of exams, and kind of relieving the stress, because you can say, look, here's this thing that someone's made. It's not perfect, but it got good marks. The problem is like it's just wrong. I mean, they've done a great punnet square, but like they've misunderstood the question. They've given the wrong response. I don't know. It feels frustrating to me. It was frustrating enough that I wanted to make a video, but I'm interested in what the wider YouTube audience thinks. Do you think that this is not a big deal? Like, in terms of writing, it's fine. There's a few spelling mistakes and grammatical errors, but for an exam conditions, it's perfectly acceptable. At this level, the student does understand what's going on. It's just like, I can imagine getting bamboozled. Like, you're up all night studying, the exam's tomorrow, you read this essay, you're like, okay, let me just understand the structure. And then you just get incredibly confused because it doesn't add up, because you're like, but hang on, but the exam question said this, but the answer said something else. And I don't know, maybe it's also that like it's the first one on the sheet. Like from the point of view of a student submitting this, that doesn't matter at all. The fact that you get a merit for the first question and an excellence for the second two, that's all good. But for a student reading this, you download the PDF and you start going. Yeah, it just seems rough, because like there's got to be someone out there who's written a good answer to this. And there's got to be some brilliant mind who's cracked this. Or there's got to be a teacher who can write out like an exemplary response for students to use as a reference. It's 1:30 in the middle of the night right now, so I should go to bed. But thank you for watching. I know this is a deviation from the videos I've been uploading lately, but that's what the YouTube game is about. I'm sorry if you were here looking for like a BKC style cutting edge commentary with some movie trivia attached. Maybe you'll find them in the next video. Yeah, if you're a student and you're trying to get through NCEA, then best of luck to you. I mean, it's hard, especially in biology. There's all this language, and you've got to memorize all these concepts, and a lot of them are much simpler than the jargon would let you know. It is possible to kind of just understand the like very basic cat is yellow because of machinery stuff, and then write a good essay answer. But it can be pretty opaque figuring out how to get there, and being given an exemplar response as flawed as this one, I think is an extra roadblock that you didn't need in your way. Thank you all so much for watching, and I bid you all good night, and if you've got mock exams coming up, I wish you the very best of luck. See ya.